Welcome to the Tree of Life podcast. My name is Joel Edford, and I'm happy to be here today with Professor John Harada. John Harada is a professor in the Department of Plant Biology. Welcome, John.
Welcome. Thank you very much.
So today I'd like to talk to you a little bit about your background, your academic journey, and some current research that's happening in your lab. Maybe you can start out by telling us, how did you end up at UC Davis? When did you get here? What was your path like towards becoming a scientist?
Yeah, so I grew up in East Los Angeles and went to Garfield High School. Garfield High School is famous, and that's because there's a movie made about Garfield High School. It's called Stand and Deliver. And what Stand and Deliver was about, it's about a AP calculus teacher named Jaime Escalante. And he was a very devoted and excellent teacher, and he taught the students at Garfield High School so well that all of them passed the AP exam. Now, the reason why that was a surprise to everyone, including the college boards, was because Garfield High School was at the bottom of the rankings of the LA City School District, and LA City School District was at the bottom of the rankings of the state of California. And so how could a teacher at a high school that's 90% Chicano, how could he teach them all to pass the AP exam? And so what the college boards did was they made all the students take the exam over again, and they all passed. So that just, I say that just to kind of illustrate where I started when I went to college. Because of the rankings of Garfield High School, I ended up being, I think, second in my class. But when I got to UCLA, which is where I did my undergraduate work, I didn't know anything. And so I spent the first quarter and year just trying to catch up. And I will always remember this because I'm a first-generation college graduate. But I'm not the first one who went to college. My older brother was. And what my older brother told me when I got to UCLA was, he said, "This is really hard, and so just be happy getting C's." And so I was happy getting C's, but then I figured out, if I studied a little bit, I could actually do better. And so from there, I didn't really know what I wanted to be. I thought maybe a medical doctor because I didn't have a good feel for what the different career options were in biology. But I took a biochemistry class from a professor named Charlie West. And it was sitting in that biochemistry class that I sort of realized that all of life is just directed chemistry. And so if you can understand the chemistry, then you can understand life. And I remember clearly going to my professor, going to his office hours and saying, is this really true? I mean, is life just directed chemistry and enzymes determine how that chemistry is going to work? And he said, yeah. And he invited me to work in his lab. And that's where I got my first taste for research. And that's where I decided that, I kind of like this research. I think I might want to continue on with this.
So I have to go. Let me pause there. I have to go back and ask you. I mean, of course, as a former high school teacher myself, so were you there when Jaime Escalante was a teacher?
No, I was actually there before him.
Okay, so you were there before him. All right. And so you never had that interaction, but you were at that high school. That's really amazing. And then that experience at UCLA where you realize that maybe you're kind of in a hole there in terms of preparation. I mean, I feel like that's a common experience. I mean, I definitely experienced that as well. But I really love this connection to research. When do you know what year you were when you started working in the lab? And do you think that working in that lab had an impact on the rest of the stuff that you were studying, including your performance and how you were preparing, etc.?
Yeah, definitely. So I think I started working in Dr. West's lab when I was a junior And part of that was I was supporting myself all through college, and so I had always had a job, and so I was working, but I decided that it was really important to try and get this laboratory experience to see what it was and if it's something that I liked. And just having that experience, learning about what research is, learning about the environment of the lab, having interactions with other students and postdocs in the lab, that really gave me a feel for what research was all about. And so that was a major point in my life in terms of the directions that I would take from there.
And your brother said it's okay to just get C's, but you were not okay with that, and so you decided with a little effort you could do even better.
Yes, yeah. So I should say my brother ended up getting his degree in civil engineering.
Excellent.
But I think he worked for about 5 years and decided that's not what he really wanted to do. So he ended up laying tile, and he's very happy doing that.
Got it. So you had that great experience in a research lab. How was the transition to graduate school?
So because of my experience in biochemistry, I continued on in biochemistry. And so I went to the University of Washington in Seattle. So I was looking for a good biochemistry school. So I talked to my professors and asked where would be, the good places to go. And University of Washington was one of those. And so I remember applying, and I was able to get in. And so as a graduate student, I worked on mammalian cell biology. So I worked on these chemicals called polyamines and their role in controlling cell proliferation. And so I worked on mammalian cell culture, did a lot of cell biology. And I got my degree. And at the time that I got my degree, people were just starting to do molecular biology. So people were first starting to clone genes. And I thought, wow, that's something I really need to get into. And I didn't tell you this, but Dr. West actually worked on gibberellin biochemistry.
Okay.
And so I had some background in plants, although I never had taken a class in plant biology. And so I thought, plant molecular biology sounds like a good combination. And so at that time, there were maybe 5 or 6 people who were doing what you would call plant molecular biology. One of them was at UCLA, Professor Bob Goldberg, and I was able to get into his lab as a postdoc. And it's there that I really learned about plants, learned about molecular biology. But of course, I didn't have background in any of those topics. And so there were undergraduates who were teaching me things when I was a postdoc. So you're always learning, and you always learn from the people who know about it.
So what? That's really great. That's really great transition there. And you get there, and you probably didn't know too much about plants. I guess one question I would have is, what do you think it is about plants that makes them good models for learning about cell biology or biochemistry?
So one thing I like about plants is they basically do things differently from animals, right? A lot of the physiological processes are different, but at the core, most of the underlying molecular principles are the same. And so you can take what you learn from animals and apply it to plants, except when you apply it to plants, they end up working in different ways to enable those new physiological, morphological features to occur. And so it's sort of a way of bringing together information that you have from animals and information that you have from plants, and learning about new pathways that control plants, and especially their development.
So you're in the lab, you, I presume, get captivated by studying plants. And what was the transition like from postdoc to UC Davis, I presume?
Yes, so At the time that I finished my postdoc, so I think I was a postdoc for 3 or 4 years or something like that. And at the time, everyone wanted a plant molecular biologist, and there weren't very many of us out there who were, ready to get jobs. And so I think I applied to 6 different positions, and I think I was made offers at 5 of those different places. And it wasn't necessarily me, but it was just I had the skill sets that they needed in order
I
to
think it
build
was
in
you
these
too.
areas. And so I chose Davis, number 1, because it's California, and I'm a California boy. But number 2, Davis had such strength in plant biology, and I knew that was my weakness in terms of my background. And so I knew if I came to Davis that I could have the expertise, I've had access to the expertise that I would need to provide basic information about plant biology. And, if I had questions about things, then often I was able to go to the world's expert and ask them directly the questions that I had. And so it turned out to be a really good fit for both of us, I think. I provided UC Davis this expertise in plant molecular biology, and Davis provided the background in plant biology, the expertise in plant biology, but it also provided a really nurturing environment for a new assistant professor. I'm always grateful to Davis because they spent a lot of time helping me to get to become a professor. So I will always be grateful to UC Davis for that.
So I want to ask you about your research. So in the department, as a non-plant biologist myself, I guess I often think of you as a plant developmental biologist.
Mm-hmm.
And so what is that? What is a plant developmental biologist? What are the active areas of research happening in your lab right now?
So I've always been interested in seeds, and there's a variety of reasons for that. So the ability of plants to make seeds, they in large part account for the success of the angiosperms. Plants that are able to make seeds, they've been able to radiate, and 90% of all plants turn out to be angiosperms. Seeds are also important from an agricultural standpoint and societal standpoint as well. So I think it's something like, it's estimated that 50 to 70% of the calories consumed by humans come directly from seeds. And you think about lots of the impoverished nations, their main staple was in many cases rice, right? And so that accounts for most of the calories that people are consuming. And so in the long term, this isn't necessarily things that I will do, but perhaps the information that I develop may be useful in developing strategies to be able to produce more seeds or bigger seeds or more nutritious seeds. But from a developmental standpoint, which is what I'm interested in, the seed is really it's just an elegant developmental system. So there's 3 major regions of the seed. So there is the embryo, there's the endosperm, and the seed coat. Each of these has a slightly different genome. So the embryo is diploid and zygotic, so 1 set of chromosomes from the mother, 1 from the father. The endosperm is triploid and zygotic, 2 sets of chromosomes from the mother, 1 from the father. And the seed coat is all maternal in origin, so only female chromosomes. But yet, in order for the seed to be able to grow and develop, the growth and development of all of those different regions has to be in coordination with each other. And so there's got to be communication between these different parts of the seed. And so what my research has been involved in over the years is to try and understand basically how plants make seeds. So we focus a lot at the genomic level, at the gene level. And so what we would like to know is what are all the genes that are required to make a seed. and what is the function of those genes. Now, obviously, you can't do all of those. You can't cover all of that. And so we've focused in, one of the things that we focused in on is a phase of seed development that's known as the maturation phase. So you can think of seed development as occurring in two overlapping but distinct phases. So there's an early phase of what's called morphogenesis, and that's basically where the embryo undergoes developmental processes that give rise to the different cell types, the different tissues, the different organs of the plant embryo.
How long is that post-fertilization?
It depends on the plants that you're talking about. Most of our work now is done in soybean, and so most of that morphogenesis phase occupies about the first third of seed development. So in soybean, that's maybe 25 days or so. And so the larger part of seed development is a period called maturation. It's during this maturation period that the seed accomplishes all the things that make seeds famous. So the embryos within seeds, they can withstand desiccation, right? At the end of seed development, the embryo is maybe 5% water content. You and I would be dead if we were 5% water content. And so the seed mounts processes that allows the embryo to withstand the stresses that occur during dehydration or during desiccation that allows the embryo to remain viable even though it's under this low water content. It's also during this maturation phase that the molecules that make seeds so nutritious, the proteins, the lipids, in some cases the carbohydrates, it's during this maturation phase that those compounds are made. And so, you take a tiny embryo at the end of morphogenesis, and by the end of maturation, it's now large. All of that growth is due to cell expansion and not cell division, or almost all of it is due to cell expansion. And that's because the cells are just filling up with these proteins, these lipids, these carbohydrates that provide a source of energy for the plant to use as it germinates and before it becomes photosynthetically competent. So the maturation phase is a very important period of seed development from a physiological standpoint, but also from the standpoint of feeding the world.
So I guess the last thing I'll ask you about is, I know that you recently retired, but it sounds like projects are still full steam in your lab. So what's next for you? What do you envision as future sort of work that you're interested in? And I'll definitely put a link to your lab website so people can learn about it. But what's next?
So what's next is to complete a lot of these papers that I have data for but haven't written yet. So I think I'm going to spend the next 2 or 3 years writing up a lot of those papers. I am teaching now. I've always loved to teach, and so the department was gracious enough to allow me to continue teaching. So I'm teaching this quarter, and I'll be teaching next winter quarter. And so I look forward to being able to keep that in context too. And then I have a number of professional responsibilities that I have. I'm on advisory committees and I'm reviewing grants and papers, so I will continue to do that. But I can kind of see how things are gradually going to decrease, at least in terms of my academic endeavors, and I think it's about time for that too.
Well, you've had an amazing career, and it sounds to me like your retirement is probably a lot more like semi-retirement, but I'm definitely grateful for your time here today and for your dedication to the campus and all the students. So thanks for coming on.
Well, thank you, and let me just say one thing. It makes it easier to retire when I know that people like you are going to carry on the traditions of the department, especially you in terms of your teaching. So I very much appreciate having you in the department to continue to increase the effectiveness of our teaching in our department.
Well, thanks, John.